came to know that yes, wood anatomy, there is a specialized lab over there. And when I gone through the basic concepts, so I found it very interesting and worth exploring. And the other important thing is that this field or this study is not much explored, at least in our country. So that itself gives you a point that there is a lot of scope available. So let us explore, right? It is not that it is not at all touched upon. There are some points which are included in the curriculum, even at the undergraduate level of all the major universities, as I have seen. So we have to understand a little bit detailed aspects. And as per the schedule, as you know, today it will be based on the introductory concept and some basic techniques, and the highlighting on the growth rings as well. So these points will be discussed, and there will be some interesting features and aspects related to this, which I will show, which I will highlight, and hope it is going to be an interesting experience for everybody. Right. So the topic, as we know, it is the science of the wood, and the word which is used over here is science, and not just focusing on botany as such. Yes, it is science because botany is one of the major parts. We are going to touch upon the botanical aspects as well. The major part will be the botanical part. But when you study about the wood, it also has got various other aspects to it. Right? So we'll see them one by one, and it will be clarified to us. So at any given time, if or maybe towards the end, like we can have an interactive session, I would like to listen from all of you so that it will be a fruitful discussion. Right? So basically, including today as well as tomorrow, the major contents of my discussion would be highlighting or revolving around these points. Like basically, I will introduce everybody to the science of wood. Okay, and what basically we'll start right from the very basics, right from the scratch, so that it was within the understanding limit of everybody. So the science of the wood, and then when it comes to wood, everybody is aware about something called growth rings. So what exactly is a growth ring? How to understand what is a growth ring? What are the different components, and what are its different applications? All that becomes extremely important. So growth rings followed by something called heartwood and sapwood. Then similarly, you have the different types of wood. Then the microstructure. This part will be focused more basically on in tomorrow's session. The anatomical aspects will be discussed. Then the different methodology, like when you have to carry out this study practically, what are the different techniques and methods that we can use, including the sectioning, microscopic observation, everything. It is ultimately a anatomical analysis. So those techniques would be discussed, followed by whatever interesting experiences which I had. So I will be sharing with the young crowd. So this will form a part of this today's and tomorrow's discussion. So when we start from the beginning, if I have to introduce this science of wood, so the first thing that can come to our mind or we can discuss is that wood is something, it's a common term, and we come across wood in our daily life in various ways. I guess everybody would agree to this, right? So wood is something which we use in some or the other way in various ways. For example, the chairs and the benches in which all of you are sitting right now, or the furnitures at your home, right? They are made out of wood. Some of them are very costly. Everybody is aware, right? So we prefer furnitures made of very good quality wood. Okay, the polished ones, right? The shining ones. So there are different types of furniture. Similarly, all of you, when you were in school, now when you're in college, right? So you sit on the benches, right? And sometimes there are lots of creativity which is shown on these benches, right? So they are also made out of wood, correct? Then, yes, those people like who are quite interested in cricket. India is a place where like cricket is something which we cannot avoid, correct? So that cricket is the one cricket bat is made out of wood. This is one example to show that how even goods for sports can be including wood, right? And then you have something called the pencil, very common one, right? So right from the childhood, you have been using a lot of pencils. You have been sharpening them, drawing with them. So now when you look at a pencil, you can observe that yes, it is made out of wood, right? And it is completely a useful thing in our daily life. It's a part and parcel of our life. Similarly, different other sports goods like the carom board, right? Especially since the last year in this lockdown, people have got a lot of time to play these indoor games at home, right? So again, wood comes into this picture, correct? The cricket stumps again, along with the bat, the stumps are also there, right? This kind of keychains, all of you must have noticed, especially if you visit any hill station. So you find those, like in the north, especially there are these mall roads in the hill stations. So you find these wooden artifacts present. So if you go over there and if you want to buy some gifts for your friends or relatives back home, so you prefer to buy these kind of keychains. Sometimes there are letters engraved on it, and all those different types are available. So they are also basically made out of wood. Okay, then you all must have noticed the railway sleepers, these ones. Nowadays they are made out of cement mostly. The cement blocks are used, but earlier there were a lot of wooden railway sleepers which were also useful. At some places they are used even now. So this itself is giving you an idea. Like when it is a railway sleeper and the huge railway train or the engine having tons of weight, when it is rattling across this railway track, the sleepers have to be so strong, right? So when you say wood, the another word that similarly comes to our mind is something called strength, correct? So it has got those physical properties, compressibility, extensibility, right? So these characteristics of the wood are the ones that help to make it useful in our daily life. And then something in the kitchen also. In some places we have those wooden spoons, wooden pans, right? They are also quite useful in the kitchen. So they are also made out of wood, and it is a natural product, right? So there are various other materials as well, but here we are looking at it from the wood point of view. So these are some of the few examples which I'm showing you, that is giving you an idea how wood is coming around us in everyday life. So when you are, what you can say, we cannot avoid wood in our daily life. It's better to know some little more in-depth idea about what is the wood, right? So let's go a little further. When it comes to wood, another correlated word that comes to our mind is the timber. Again, it's not a new term for everyone. We know what is a timber, right? When we saw the different examples where wood is used in everyday life, another point that must be remembered is that you cannot use any species of wood for any purpose. Wood is a completely naturally made tissue, but at the same time, it varies across different species. So we cannot use any wood species for making a furniture or a railway sleeper. The requirement should be the criteria should be our requirement, the strength. So that's why the word is timber. There are some species of plants where the wood which is produced is so strong and they are extremely costly also. So they are commercially useful. Such wood are called as timbers. So all woods are not timbers, but all timbers are woods. So ultimately, when you say it's a timber, it means the wood of such plant species which are commercially useful for various purposes. So in India, okay, looking at the basic ecological aspect of biodiversity in our country, when you look at the biodiversity of woody species in our country, we know very well that we are abundant in that. India is such a country which is blessed with a huge biodiversity of shrubs and trees belonging to different groups in the plant kingdom. So ultimately, if you ask, if there is a question in the mind that what scope this kind of a study has, so the biodiversity itself is giving you the answer. Okay, from north to south and from east to west, we have immense diversity of the tree species. That means we are having a plethora of woody species in our country in all different forms. Especially if you compare with the other countries of the world, especially in the Western countries, there are more of the gymnosperm species which are present. But in our country, we have gymnosperms to angiosperms to like every other species, right from pteridophytes onwards. So let us take a look at some few examples of some major timber trees. Right, this is something Shoria robusta. I hope everybody is aware about the botanical names and these common plants. Right, Shoria robusta, commonly known as the sal tree. Now, this is one species which is very abundant in the means towards the eastern part, in fact, like means all the way to Jharkhand, Orissa, north Haryana, Uttarakhand. This tree is very abundant, and they grow in huge densities. So this is one species which produces very good quality timber. That's called Shoria robusta or the sal tree. Similarly, you have this Deodara tree, Cedrus deodara. This is a gymnosperm species. So this one grows in the high altitudes of our country. In the higher reaches of Uttarakhand, Himachal Pradesh, you will find this Cedrus deodara growing, and it produces very good quality timber. Then you have this one, Holoptelia integrifolia. This one also produces very good quality wood, and when it comes to wood, there is a property called as a grain, wood grain. It's like morphologically when you look at the wood, so there are some kind of a striations which you observe. That's called as a grain. So this Holoptelia integrifolia produces very good quality grain, and the association of the tissues in this wood is so strong that it makes a very good timber. That is also abundant in our country. Then this is the most common one. In any hill station, if you go, you see mountain sides full of pine trees. The Pinus species, having various Pinus genus, various species are abundant. So that's a very important timber species in our country. And then you have this one. If you come down to the plains and the deciduous parts of our country, the tropical and the subtropical regions, so you will find this Tectona grandis. That's nothing but the teak wood. And teak wood, a pure quality, a fully grown one, is very, very costly and expensive timber. This is one of the plants which even farmers used for agroforestry purposes. They grow it around the agricultural fields and they gain additional income because of that. So this is Tectona grandis. Here in Mumbai, near Mumbai, as all of you must be aware that we are closer to those Western Ghats. So if you go towards from Mumbai, if you travel towards Pune, we come across those Khandala and Lonavala Ghats. So there the mountain sides are full of these teak trees, Tectona grandis, and you can identify it very easily by its inflorescence. The wood is very nice, ring porous wood. Internally also, the wood has got a very beautiful architecture. Okay. Then this is Taxus, again a gymnosperm grown in high altitudes. It's commonly known as the yew plant, YEW, and it produces very good quality timber. Among the softwood, this is one of the best. And another example I can give is, yeah, that is Dalbergia sisu, the shisham plant. And shisham is also a very good quality timber. Shisham, and there's another one called rosewood, which is nothing but Dalbergia latifolia. So these two are different species of the same genus, that is Dalbergia fabaceae or leguminaceae member, and produces good quality timber. So there are very good woody species even among the legumes. Okay, so these are some of the common species which are used for timbers and which are found in our country, which I have highlighted as beyond this in fact. So the list becomes endless. Okay, so this itself is giving you an idea when it comes to wood and timber, how important it is for our country and to understand them, right? So it automatically leads to the point that we must be conserving such species too. Okay, going on to the next one. So having this background that yes, wood is a part of our daily life, we use it for so many different purposes, right? It is associated with timber species, like we use it for furniture, construction, etc. So now coming to the botanical aspect, basically what is wood, right? So when you say what is wood, that's the common term that we use. But in botany, technically, the wood that you understand is nothing but the secondary xylem. In anatomy, people study about the basic plant tissues, where we are aware about something called vascular tissues in plants. And in the process of plant evolution, right from thallophytes, algae, all the way to the monocots, the evolution of the vasculature in plants has been a very significant process. All of us are very well aware that algae gave rise to bryophytes, which ultimately developed to form the pteridophytes. So this transition, especially from the bryophytes to pteridophytes and pteridophytes to gymnosperms, has been a huge transition, because this is where the plants invaded the land very properly. Before that, plants were aquatic and they were very close to the ground. But now, because of these vascular tissues, especially if I'm not wrong, during the Carboniferous period in the geological time scale. So what happened was that the plants developed these vasculature to a great extent, because of which it became possible for the plants that they can absorb the water from the soil and they can transport it against gravity all the way to the higher parts of the plants. And that's why, if you notice in this plant evolution, the pteridophytes and especially the gymnosperms, they are some of the tallest and the largest plants on the earth. So why they could afford to grow so tall, unlike the bryophytes or the algae? It is because of this vasculature. So it's not just about the height. When a plant is growing so much in height, in order to support such grow in girth. So the height and the girth both are very important. And that was possible because the plants developed something called cambium tissue. Cambium tissue, as we all know, is present between the xylem and the phloem. The primary vascular tissue is present in every plant, the xylem and the phloem. But as and when the plant increases in size, the need arises that the girth must increase. And it is the cambium tissue that gives rise to secondary xylem and secondary phloem. That secondary xylem which I am talking about is nothing but the wood. So ultimately, if you say what is wood, it is nothing but the secondary xylem of mature shrub. The wood is not applicable for herbaceous plants. They do not have any wood. They have a stem, but not a wood. So for shrubs, trees, and lianas, lianas are nothing but woody climbers, as you can see in this picture. Okay, Entada is an example of a liana. Tree species, there are many of them. Shrubs, there are many of them. So they produce these kind of woody tissues. So here, as I said, this secondary xylem, which is the wood, it is produced as a result of secondary growth. So in the syllabus, in the curriculum, all of you must have learned about something called secondary growth. So it's like the cambium tissue is present. I'll show a diagram after this. So the cambium tissue will divide and it will produce the meristematic portion of the cambium will produce secondary xylem as well as secondary. So one can admire this, how beautifully during evolution the planning was there. Yes, the plant should increase in height so that it is able to invade the land. And when during evolution, when the plants initially were living in the water and were surviving so nicely, it was able to access the water so easily, so there was no need of vascular tissue as such. Similarly, when it came to the land, initially it was just a small thallus directly attached to the substratum. So it's like you're staying in the ground floor of a building, so you don't need a staircase or an elevator. But then the building increased in size. So there was a need. Yes, there should be some staircase on an elevator, and that is provided in the wood. So that the roots can grow deep into the soil, absorb all the requirements, pass it on to the topmost leaf in the topmost branch against gravity. So during evolution, this entire planning was possible because of the vascular tissues. And that is the secondary growth. So you must have seen many such tree species which has got such huge girth, amazing huge girth. So that is possible because of cambium. There is more of secondary growth which is produced, right? So ultimately the wood, okay, when it comes to wood, of course it is a hard structure and it is a mostly durable structure, because if there is one point of difference between the growth in animals and growth in plants, so the growth in the case of plants is continuous till the end of the life. There are, you must be aware about, there are some plant species which grow for thousands of years. Like you have those sequoia, or some of the quercus, or some of the pines. They grow to thousands of years. So when it has to grow for thousands of years, standing at just one place where it started by chance, so it has to sustain itself for that entire duration of life. So the wood must be such an efficient structure. It must be attacked by so many pathogens, so many pests. There must be so many contaminants coming from the soil. So the plant has so many mechanisms to avoid all the harmful things from going all the way to the leaves and disturbing the physiology. So the wood is the one which connects both. That's why it is a very significant structure, and it's a durable one because there is no replacement as such, right? And yes, now this, when it comes to wood, wood, as I said, is science in itself. So there is one branch of it where one can also study about fossil woods. I'll be showing some pictures in a few slides away. So fossil woods is also a very interesting aspect of this study. All of you are not aware about what are fossils. Like this branch of botany, we call it as paleobotany. So we come across so many fossil groups of plants. Some of them are preserved as wood species, some old ones. So we come to know such a great deal of idea about the plants of the past, adding to the story of evolution. So there are botanists to study this aspect as well. So we have a very good institute in Lucknow, Birbal Sahni Institute of Paleobotany. So this can be one part of it, right? And yes, as I said, during the geological time scale, the evolution of the plants had taken place, and based on the understanding till now, that the vasculature developed in the pteridophytes, and then it increased. So, if you see very tall plants, pteridophytes are slightly shorter, but if you go to gymnosperms, they are very tall. So, in the tall plants, right from the roots all the way to the leaves, it's not possible to have just one tertiary element. Tertiary element means like the conducting element, on a simple term. Conducting element of the wood which is transporting the water and the sand. So shortening of the tertiary elements means what exactly happened? Like instead of having one or two long ones, they were divided into shorter ones. So that the conduction becomes efficient. Whenever you study about adaptations, naturally inherited adaptations, all the adaptations are always for some purpose. It must have happened that initially the tertiary elements are longer, but conduction was not efficient. So over a period of time, across the period of generations, the shortening took place. And now what we have is very efficient. So this is the ultimate process of evolution, which you can also find in the case of woody tissues. So this was a very technical background about what is wood. The detailed aspects, the story is always endless, right? We'll see a little more on this. So a quick glimpse at something called secondary growth, and this is very much a part of the curriculum everywhere. So everybody must be aware about this diagram, right? Some of the students, they get nightmares when it comes to this kind of diagrams. I can understand, right? So here it's like see, there is something called as a primary vascular bundle, which is there right in the center. This you will be able to find in the case of small plants or seedlings. Okay, so there is something called cambium. Cambium is present between the xylem and the phloem. Now this cambium has got meristematic activity. Okay, so when it has meristematic activity, what happens is it produces secondary xylem towards the inside and secondary phloem towards the outside. So the one shown over here in yellow is the secondary xylem, and the one shown outside is the secondary phloem. So that's the technical word. Secondary phloem grows, it keeps on increasing in size, and it leads to the development of the cork also. So for that, there is something called cork cambium. So cork cambium gives rise to the external structure protecting the entire stem or the trunk, which we call as the bark. So right from secondary phloem to the cork is the entire bark portion. And internally, the secondary xylem is the wood portion. That's how the entire structure of the stem or the trunk is modified. So by now, I think everybody is clear with the fact that when you say secondary xylem, it is the wood. And secondary phloem to the cork portion is the bark. That's how the growth of the plant takes place. Okay, so this diagram was shown just to reflect upon that part. Okay, so okay, a quick look at something called the bark also. Now, when you say bark, though it is not exactly a part of the wood, but it is associated with the wood. You look around, you will find so many trees and shrubby species. If you go to the plant, if you observe the stem or the trunk, we see that it is covered with a bark. Some of the barks of plants are even medicinal. Cinchona bark we are aware of. So it's a peeling one. So when you say bark, why I'm focusing on that also is that because no doubt it has got its own significance. It's a covering, so the main function is protection. But at the same time, to some extent, that is useful even for species identification. May not be up to the species, but at least to the genus level. The structure of the bark is also very important. If you look around us, especially what I have seen among the students is that there are two common species which have grown most commonly in our roadsides in the city areas. Now, one of them is the gul moher, that is Delonix regia, and the other one is son moher or copper pod that we say, that is Peltophorum pterocarpum. These are the two very commonly grown in our cities in the roadside. So though we technically see these plants everywhere, but even for students within the fraternity, the two species create a confusion, especially when they are not in a flowering stage. Both of them flower during the summer months, in the months of May month, April and May. They are nicely in flowering. So Gulmohar, you know very well, they produce nice bright red colored flowers, and Peltophorum produces yellow flowers. So when they are in a flowering, it is easy to identify. But when they are in only in a vegetative stage, when there is no flowering, how to identify? Yeah, they are not very similar. Yes, from the leaves, those who are experienced, they will identify easily. But from the student's point of view, I'm telling that the identification becomes difficult. So that's the time when you can look at the trunk and the bark. If you observe closely, the Delonix regia, that is Gulmohar, the bark is very smooth. And for copper pod, that is Peltophorum, the bark is rough and it has got those striations. So these are some features generally, most of the time we tend to avoid or ignore or sideline. We look at the leaves, we look at the flowers for identification. Sometimes the trunk, the stem, the wood or the bark can also be features which are useful for the identification. So based on that, let me show you one more picture where we can observe, you must have seen, but you may not have observed much, the different types of barks in plants, right, on the trunk. Okay, these are very few examples which I have shown. There are many others which you can include, like just the types on a broader scale. You can just take a look. Like some of the plants, they have very smooth bark, smooth ones. If you see the bark of say Polyalthea, Ashok, false Ashok, right? Or there are barks which show the presence of lenticels. Anatomically, we study about this. That when the cork cambium it opens up, it produces an opening called the lenticels during the secondary growth. So a part of the transpiration also takes place from here. So in some of the plants, the lenticels are very prominent in it. If you see the bark of say Leucina leucocephala, the lenticels are prominent. If you see the bark of Anthocephalus, again they are very prominent. Elstonia scolaris, very prominent lenticels, white colored ones. Okay, peeling strips, like you can just peel off in small parts. Okay, or vertical cracks. Okay, or sometimes they can be horizontal cracks. So you should be aware about the difference between cracks and scales. So that has to be observed carefully, right? then sometimes in the form of plates, like big pieces, right? So vertical strips can be there, intersecting ridges or ridges broken horizontally. Intersecting means like networking pattern, or ridges broken horizontally means very fine striations, like, or there can be uninterrupted ridges. So we can always venture into the details of all this and explore more. But this is the introductory part which I wanted to show all of you. And there are respective examples given. You can, maybe what you can do, like, the best part about this kind of study is that you can always look around and try to identify when you know the types. Morphological feature, you just need to look at the plant, stare at the plant, and identify, okay, this what kind of bark it must be, and try to identify. That's a very interesting study, right? So this is about the bark. And yes, the bark is extremely important for the wood also, because it protects. See, when it comes to wood of trunks, wood of trees, the two major damaging features for the wood are, one of them is fungal attack, and second one is attack by pest, like insects. There are insects like stem borers and all, which can damage the wood like anything. They can damage like anything. I heard one thing, especially in the 1990s. It happened in one of the forests of Madhya Pradesh. More than 9,000, I mean, sorry, 9,000 or maybe 90,000. The trees were simply damaged just because of the attack by borer insects. It happens. So they can cause a great deal of damage. So the bark is very important protective feature. If you see the bark of trees like very common Ficus bengalensis, the bark is very thick. Even in the case of Azadi Rakta, neem tree, the bark is quite thick. But if you see eucalyptus, everybody must have seen. The bark is pinkish in color and it can come off in strips, thin ones. So the bark is a very significant feature. A lot of some variations you can find in this bark. So this is also an example of diversity of different features that you see, right? So nature has provided that. See, these are the examples of fossil woods, okay, archaeological specimens. So one can understand this can become a very important interdisciplinary study of botany with archaeology. And always remember, in case of career point of view, if you do something interdisciplinary, it becomes extremely interesting. There are some research papers available where people have studied this kind of archaeological specimens for their botanical aspects. Like in the ancient times, maybe from some tribal regions, they could excavate some of these kind of masks and all which people used in those times. Or maybe this kind of mortar pestle which people used in those times. They were made of wood. So when they are founded, in the case of excavations, the archaeologists need the expertise of botanists and wood scientists in order to understand the specimen which it must be. Because for example, say mortar pestle, something has to be grind on it. That means the mortar has to be a strong one. So the species, one of the papers I saw, this kind of a mortar pestle was examined, and it turned out to be from Ziziphus jujuba. And we today also we know very well that it is having a one of the very strong woods. So people were aware about it that time. So it just adds to this kind of interesting knowledge, right? So fossil woods, archaeological studies can be really very interesting features, and there are really good research papers available on this. Okay, so that can be one interesting option if you are interested in two different subjects like this. Okay, so based on this background, by now I think I can tell clearly, like when you study about wood, what are the different interesting aspects. Yes, it is a very important part of higher plant structure. Higher plant in the sense, the vascular plants, right? The efficiency or the significance of this part cannot be excluded, right? And this is a very important point. I mentioned that the anatomical features of the wood are highly conservative for a species. We need to remember this point even during tomorrow's session. When I say it's a highly conservative feature, the meaning of this statement is that um, taxonomically, like there are diverse fields under taxonomy which are coming up these days. And the basic meaning of taxonomy is that looking at the plant features, we identify the genus and the species. So generally, or most often, the reproductive features of plants are taken into account and we arrive at the conclusion that it belongs to so and so species. But one should remember that wood anatomy or the entire structure of the wood is also a very important feature which is highly conservative. When you say highly conservative, it means for a given species, the structure of the wood remains constant, the internal anatomical structure. So here, let me tell you one more point that the anatomical structure of a wood is not very simple as it seems. Tomorrow I'll be highlighting more on the anatomical aspect, but here let me tell you one more point that in a given wood sample, if I am talking about a angiospermic wood, in a given wood sample, there are more than 250 features to be studied. This is for reference. So the number of features go to more than 250. The features are different for gymnosperms and for angiosperms. So when you say 250 features, quantitative as well as qualitative, both of them together for a given species has got a fixed structure. Even for the quantitative features, the values which we get is within any given species. That's the reason we say it is highly conservative. The features won't change. And that is why many times it happens that any random wood sample is given and looking at the structure, cross-checking it with the available literature and reference, you arrive at a conclusion that this is the species. And that is authentic. If you are a wood anatomist, if you are a researcher in this field, you have the authority. That's the interesting part of this. It's a highly conservative one. So obviously it has got a lot of applications. As I give the examples now, that when you study about wood, you get a lot of applications in other fields, taxonomy, as I gave the example, phylogeny, the evolutionary history, right? Even the plant ecology, obviously, right? Because the structure of the wood is determined on the basis of the environmental features also. Because there are a lot of adaptations which have been developed over a period of years. And then it becomes a species character. So environment also plays a role, and we can associate it in so many other different ways. Archaeology, I gave the example, right? To a large extent, even plant pathology, plant entomology, in fact, right? Everything can be associated with wood. So it's an interdisciplinary science. And wood has got a lot of practical applications in other numerous fields, archaeology, even sports manufacturing, furniture, construction, railways, etc. So it has got a lot of importance. And as I said, there is a good diversity of species of commercial timbers in our country. It is very large across the length and breadth of our country. So that itself gives you an important scope to immense research in this Indian scenario, right? So that gives you an idea that yes, if I take up wood anatomy or wood science as a subject, there can be a very good career one can make in it. And there are people required to work. A very small number of people. I had the opportunity to work, but as a student, as a PhD, later on I entered into teaching field. So I'm touching upon everything, but along with that, as I'm doing right now, I'm sharing this idea with everyone, so one can get an idea. Because even I was one, right? Let me show you some more interesting aspects. Yes, well, little insight. People of you must have seen that many times on a truck or a tempo, the logs of wood which are placed and they are being taken somewhere, maybe to the industry or where. So you see the logs, the observation or a structure see inside, let's start from the outermost part. So there is the bark, first of all, an outer bark and an inner bark. So imagine the level of protection given to the plants. Thickness or the size of the bark can vary. It also depends upon the age of the tree. Okay. Internally, broadly, if you observe, you see that the central portion of the trunk, this one, is slightly darker in color as compared to the outer lighter portion. These two regions in a wood are different. The inner central darker region is called as the heartwood. The heart is in the center, you can remember it that way. So it is called as the heartwood. And the outer lighter colored portion is called the sapwood. We'll see the differences between them now. Okay. And then in the heartwood as well as the sapwood, starting from the centermost portion, that is the pith, towards the periphery, you can say concentric ring-like structures. They are known as the growth rings. So if you take a section like this, we are looking at a cross section. And if you see it perpendicular to it, we are looking at the longitudinal section. So these are the broad features that you are able to see in a trunk. The pith, the heartwood, the sapwood, and the growth rings. Okay, I think this is clear. So let us see the differences between the heartwood and the sapwood. As we have seen right now, the heartwood is the centermost portion, generally darker in color, and the sapwood is the outer lighter portion. This is applicable for all the woods. Okay, so we are technically focusing more on the gymnosperms and the angiosperms. So the differences you can see, heartwood is also known as duramen, and sapwood is also called laburnum. Okay, so the heartwood forms the central wood, and the sapwood becomes the outer portion, the outer part of a mature stem. Okay, the heartwood is generally dark colored because that is the portion where there are deposits produced as a result of secondary metabolism. Okay, the heartwood which is present now was once a sapwood. As and when the growth increased, the sapwood became heartwood in the center, and the outer portion is the sapwood. So the innermost portion of the sapwood is turning into heartwood as and when the growth is increasing. And that's why the sapwood is comparatively lighter in color. The word is sapwood. So when you say sap, it means the conducting part, water with the dissolved minerals. So that is conducted only in the sapwood, not in the heartwood. The heartwood is not involved in conduction. They are plucked. And that's the reason the heartwood is comparatively heavier in weight, and the sapwood is comparatively lighter in weight. If you see the specific gravity or the density of the wood. Okay, so the heartwood generally does not have any living cells present in it, and the sapwood has got living cells like xylem parenchyma. Okay, and it is the dead and the non-functional part having deposits giving mechanical protection, and sapwood is the functional part involved in ascent of sap. So these points are making the things clear. What is the difference between a heartwood and a sapwood? Okay. Similarly, now let there not be any confusion. You say heartwood and sapwood together. Similarly, you say hardwood and softwood together. Both are different in meanings. You say any wood, it will have heartwood and sapwood, but the wood itself is of two types. It is hardwood and softwood. So what are these basically? When you say softwood, they are literally not soft ones. In fact, they are very hard, but they have been given the name as softwood, and they are the ones which are derived from gymnosperms. Now, among gymnosperms, the major group are the conifers. So the conifers mainly form the softwoods, and they are producing very good timbers also. So here you have examples like say pinus, taxus, cedrus, araucaria, so many others. All these come under the category of softwood. I expect everybody to remember this, because tomorrow when we study about the microstructure, we will be studying the detailed microstructure or the anatomical aspects separately for a softwood and separately for a hardwood. So that time the confusion must not be there. We'll see the differences also. Okay, so that's the softwood. Generally, gymnosperm trees look like this. So simply speaking, a softwood is nothing but the wood which is obtained from gymnosperms. Similarly, by now I think the guesswork would be easy. The hardwood is nothing but which is obtained from angiosperms. And mostly it is the dicots, because in the case of monopods, there are more of the herbaceous ones. But we consider all angiosperms as hardwoods. So it is the shrubs, the trees, and the lianas. So we have examples like Betula, Shorea, Dalbergia, Tectona, Quercus, etc., and many others are there. So all those come under the category of hardwoods. So with this introduction, let us also quickly take a look at the hardwood and softwood on a larger scale. Okay, this will be shown again tomorrow. But when you say softwood and hardwood, okay, just the anatomical features, like the major difference which you will find between both of them is the presence and the absence of a very important tissue in wood called the vessels. A hardwood has got vessels, softwood does not have vessels. We are talking about xylem vessel. Here in the case of hardwood, if you can notice, there are these pores which you can see in a cross section, these ones. This can be seen only in a cross section, large and small ones. They are labeled as the vessel. So these xylem vessels are seen only in a hardwood. And in a cross section, they appear as these pores. That's the reason a hardwood is also known as porous wood, because they show the presence of these pores. But in a softwood, there are no such pores seen. The xylem vessels are not seen. You can see there are resin canals present. Canal and a vessel are two different things. Otherwise, there is no pore. So that's the reason a softwood is also called as a non-porous wood. So these words need to be remembered. Like hardwood, sapwood we have seen. Hardwood and softwood we have seen. So hardwood is also called as a porous wood. Softwood is also called a non-porous wood. I think these points are clear. There are other features of the wood also we will see in detail tomorrow. Like we have the xylem parenchyma in two different forms, axial parenchyma and ray parenchyma. Then we have the fibers, the tracheids. Okay, all that will be seen and discussed in detail. Okay, so here, as I said today, like with this introduction about the wood, we will also go to the part of the growth rings. Right? So everybody must have seen growth rings and heard about growth rings. And there's one point about the growth rings which everybody is aware, that the growth rings are the ones which are counted and you come to know the age of a tree. That's completely correct. But when it comes to growth rings, it's not just that. Counting the age is correct. But is it just about counting the age? No. There are many aspects to it. Why is a growth ring produced? How is it produced? What other things do they signify? These are the points also we can see, right? So technically speaking, when you see growth ring, okay, in a section of a wood, we find it in the form of concentric circles like this. Each circle we call it as one growth ring. That's why it is also known as an annual ring. But a single growth ring again consists of two parts, because one growth ring is completely produced in 1 year of growth. And in one entire year, approximately or broadly speaking, there are two seasons. One is a season where there is good amount of moisture that we call as the summer and the monsoon. And the other season is the dry season, which we call the winter. So during the winter, the growth is different. During the summer, the growth is different. Both of them together is forming one growth ring. So the growthing is produced as a result of seasonal differences. Let's see about that. Right. So ultimately, they are nothing but closely spaced rings in a cross section, right from the center to the periphery. So the one which is there in the center, the wood might be very huge, but the one which is found in the center is the first growth ring of the plant's life. And the one found towards the periphery is the last growth ring in the plant's life. Right? So that's why they are also known as annual rings, because one growth ring produced in 1 year. And as I said, a single growth ring consists of two components. One of them is called the early wood, also known as the spring wood, and the other one is called late wood, also called the autumn wood. So now again, hardwood, sapwood, hardwood, softwood, early wood, late wood. So there are confusing terms, but it's not too much of a confusion, I suppose. See, early wood is also called spring wood. The word spring is nothing but a season, and spring is considered to be a favorable season, having good moisture and all. Similarly, late wood is also called autumn. Autumn is another season, considered to be nearing the dry season. So why we are talking about seasonal moisture availability? Because in a wood there are important conducting elements that transport water. In case of gymnosperms, it is forms, it is the vessels that do the work. But conducting elements are present. So when the wood is being formed during the spring season, can be broader in size because more water can get transported. But when it is an autumn season, when the water availability is less, the conducting elements will be comparatively smaller in the girth or the diameter, or maybe the difference will be in the wall thickness. So in the same wood, if you go microscopically and observe, the spring wood will have larger vessels or conducting elements, and autumn wood will have smaller vessels or conducting elements. That's the reason both of them appear different. So that's how you can differentiate between early wood and late wood. Similarly, there are sometimes what happens is in any particular region when the plant is growing, a particular climatic event can take place. Maybe there are floods or droughts or any hurricane or anything like that. To some extent, they are also reflected in the growth rates. There is a disturbance in the growthing produced at that region, and that has been studied scientifically. That in a large wood sample, in one of the growth rings, there is some difference observed, and when it is matched with the meteorological data, it comes out that that particular year there was some climatic event. Disturbances reflected. May not be every time, but that is one clue we get. Okay, so that's the reason it is said early wood vessel resembles the wood of a tropical rainforest tree because it is getting moisture continuously. But a late wood vessel resembles the wood of a desert shrub where the water availability is scarce. So an early wood and late wood together is forming one growth ring, ideally. So that's the reason the width of the growth ring has been directly correlated with water availability. If the water availability is good for the major part of the year, generally the growth ring is thin or smaller in diameter. If the water availability is difficult, then the growth ring is small, or vice versa, according to the water availability. Right? So species which are deciduous. Deciduous ones generally grow in such places naturally. I'm not talking about the cultivated ones. Okay, naturally the species grow in such regions where the seasons are very distinct, summer and winter. Unlike in a city like Mumbai, where we are. Here, what happens is throughout the year, more or less the climatic condition is similar, not much difference. And nearby areas also. If you go to the evergreen species, they receive water moisture continuously throughout the year. So there the growth rings are very indistinct. You cannot make it out with the naked eye. Though the growth rings are technically produced, but they are very similar. So growth rings are able to give you an idea about this also. And then the main point which everybody is clearly aware, that growth rings, when you count, you are able to get the age of a tree. That branch of studying growth ring is called dendrochronology. That's again a very interesting aspect. There's one very interesting photograph I'll show you towards the end. Okay, so the fact that growth rings are extremely significant for various aspects. They show the transition between seasons. They are giving you an idea about the geographical distribution of plants, the phytoclimatic condition, the amount of the water availability in different seasons, right? And obviously, the counting of the growth ring is giving you an idea about the age when the plant died, right? Or any abnormal climatic event, as I mentioned, any abrupt or any abnormal condition is reflected very nicely in the growth ring. So that's one other significance. And sometimes taxonomic character is also hidden in the book. For example, this one, in order to distinguish between very closely related species, okay, like for example, Ulmus americana and Ulmus rubra. The difference is there in the growth rings to see. Ulmus americana has a growth ring, an early wood zone, which is just one vessel deep, while Ulmus rubra has an early wood zone which is more than two vessels deep. So the number of vessels considered in ideal condition can become a very important character for distinguishing between species. It's a taxonomic feature, right? And then obviously we can correlate growth rates in fossil woods, especially for such species which have become extinct. So growth rates give you a very good idea about the diversity and the distribution of such plant species in those times as well. So the study of growth rings is extremely important. It's the most important part. Okay. So the introductory part is done, and we have discussed about the growth rings also. Let us take a look at the different methodologies and the techniques when it comes to wood science from the botany point of view. See, when it comes to studies on wood, if you have to do it practically, the two main things that you require is that three main things, in fact, you can say. The first thing is how to collect the wood sample. See, collecting the leaves from the plants is comparatively easy. Even if it is a tall plant, if you have something to access the leaves, you can just pluck it off and carry it with you. Yes, there are techniques associated with that also, herbarium and all, right? But if you are in front of a big tree, and if I want to collect the wood sample out of it, of course, it is not advisable that you will cut down the tree and then take the wood with you. So how do you collect the wood from a huge tree? So here, let me tell you one more important, or two words important over here. One of them is called mature wood. Other one is called juvenile wood. See, juvenile wood means it's a young wood, which you might find. You might say that collection of wood may not be possible from a trunk. So I will pluck it from a branch. Yes, it is easier to break down a branch and then study the wood sample. But a branch is a younger one. The wood is not fully formed. So the features which you will get in a branch will be of a juvenile wood, which may not be the correct one for that species because it is still in a growing stage. So if you are doing it for authentic identification, you have to take it from the mature wood sample. So from that's why the very old branch or the trunk becomes important. A wood sample from a trunk is carried out, right? This is the trunk. You are in a forest or any such place. You are in front of the plant. Okay, you are standing next to the trunk. Okay, considering that it is easily accessible. So the first thing that you need to do, as based on the structure, you must have understood by now that the outermost covering is the bark. So you need to remove that so that you can access the wood. So that can be done by very simple instruments like this. Okay, hammer and a chisel. You can just remove small portion of the bark. Let it remain as small as possible, because sometimes what happens is we botanists become the greatest destroyers. That is very important. We should be protecting those species also. So as small as possible, we should be removing that small portion of the bark. Okay, in a fully grown tree, that is done at the DBH level in a forest. So you will cut open the small portion of the bark so that the wood is exposed like this. And then there is a very interesting instrument which is called the increment borer. This instrument is used in order to collect the wood sample. Okay, these are my pictures. I can show you how I collected the wood sample from the bark. Okay, so here you are standing in front of the tree, the bark is removed. You need to insert the borer, this end of the borer, into the woody portion. And you have to hold the bars like that. It's like a steering wheel, you can say, or a cycle handle. Okay, you need to put it inside and then you need to rotate it. You can see there are these grooves present at the tip. The grooves will help to slowly it will move and go inside the wood. It appears easy, but it is extremely hard work, because it is the wood that we are talking about. The plant has made it so strong, it will not allow anything to insert so easily. So you need hard work and strong jaw muscles for that. You need to insert it, move it around, go to a little depth. Of course, you cannot go all the way internally and come out from the other side. That's beyond imagination, right? So you need to go a little deep, up to a few centimeters. And once it is fixed inside up to a few centimeters, the same way, if you initially you were moving clockwise, now you have to move it anti-clockwise and take it out back again. So when you take the, if it is done properly, carefully, and when you take it out back, this is what you will get. There will be a hole created, of course, but in that you will find small wood discs which has collected. It will be in the form of small tablets. That means from the entire huge tree, you have collected outer core small tablets of wood. Now there can be a question. The tree might be hundreds or thousands of years old. I am collecting such small samples out of it. How can I make sure that the study will be applicable for the entire tree? So one can give a thought. One can give a thought to this, that if I am taking even such small samples, like apparently it might be say around 5 centimeter each in diameter. But that 5 centimeter is having internally so many growth rings, because the distance between the growth rings is in just millimeters or even less than that. So 2 or 3 centimeter of the water sample is giving me the idea of so many growth rings. That means so many years of growth of the plant. So that means that data will not be wrong. This much wood sample is more than enough. But if you are on the field and you have done this, should I leave the plant as it is and move away? That's a very important point. Because as I said at the beginning, the two main damaging factors to the plant is that fungal attack and pest attack. If I'm leaving a hole like this, it becomes an open invitation for both of them. And that must be avoided. So for that, there are organic mixtures available. Called epoxy mixture as one of them. Okay, one can carry it along with that. And once the sample collection is done, you need to fill up the gap with those organic mixture. And initially, the bark portion which you removed again must be plugged back inside, so that you are just sealing it. Then it is not damaging to the plant. This is extremely important. Any organic mixture can also do. That is very important. So once this is done, you ensure that the plant is not damaged. And then the wood which you get like this, okay, it might be moist or wet, so you need to dry it completely. And the rest of the thing, like see, if you're collecting wood sample, it's advisable to collect even the twig sample for authentic identification in the herbarium, just to be on the safer side. Because if you are on the field, maybe you have ventured out some forest area in a far away place or something, So when the plant is in front of you, it's always important to collect all possible data, including the herbarium data. And nowadays we have the GPS coordinates that can be noted down. The preliminary data of the plant can be collected. Suitable photographic evidences must be collected, so that you don't need to go all the way again for any important point. And then preserve the samples properly. Once it dries up, appropriate labeling, it can be kept in pouches and collected back. And then once coming back to the laboratory, you need to cut the blocks in the form of this cube shape, which is called as a true wood block. This is the one which is used for sectioning purposes, because you are able to get the sections exactly in 3 different angles. I'll show you the sectioning part. Okay, so these are the true blocks that we got. Okay, and if you take it like a cylinder, for a case of a wood sample, remember the sections can be carried out in 3 different angles. Basically, there are 2 which everybody is aware, that is transverse section and a longitudinal section. Okay, transverse section is one of them, TS. We also call it as a cross section. Perpendicular to that is our LS. But the LS is of 2 types. One is RLS, radial longitudinal section. Other is TLS, that is tangential longitudinal section. If you see the pitch and considering it to be a circle, you pass through a diameter passing through the center. So if you cut the wood into half and take the section from the diameter region, LS, that is called as RLS. Similarly, this is tangential. In a circle, you know what is a tangent. If you pass a line passing through the perimeter, periphery. So here at the end, towards the end when you take a section, that becomes your TLS. This way you can say. This is the TS, the cross section. This is the centermost section which you are taking, that's the RLS. And from the periphery when you take, it's the TLS. So here today I am showing you what are the 3 different sections. In tomorrow's slide, I will be showing you how the anatomy appears different across 3 of them, and why it is necessary to study the section in all 3 angles. Okay, I hope till now it is clear, right? Then there is a instrument. Now see, when it comes to sectioning at the college level, we have been taking a lot of sections in our undergraduate and right from the before undergraduate level as well. So you normally use a sharp blade. You hold the material in your hand and then you take a section. So there is a lot of luck factor associated with that. You need to be very skillful. The blade has to be sharp. And the major thing that happens is that sometimes along with the material, you tend to take a section of your finger. So it's damaging. Many students, they are not able to do it properly. But for college level, for understanding level, that's the best thing we can show. But at a higher level, in the industrial level, if you have to take a section like this, you cannot hold a wood piece in your hand and take a section with a blade. It's not feasible for the industrial level. So for that, we have this instrument called the sliding microtome. Microtome is the instrument which is used for taking sections. There are 2 types of microtomes. One of them is for the hard tissues, like the wood that we are seeing. Other one is for the soft tissues, maybe for a plant material like leaf or a petal or something. That microtome is totally different. Here we'll focus based on the wood. That's a microtome for hard tissues. We call it a sliding microtome. This is how it works. During my PhD, I had access to this microtome for my sample sectioning purpose at the botany department of Maharaja Sayajirao Gaikwad University, that is at Vadodara, Gujarat. I had taken all my samples over there, stayed over there for about 5 to 6 months, and did the sectioning from this instrument. But during that time, because of some common contact, as Dr. James Rajput sir, who is in charge of this laboratory over there, plant anatomy laboratory at Vadodara, I came in contact with him, and it was known that this new instrument was procured at that time, a few months back. So the latest model was available. This is called the Richard sliding microtome. So when this kind of a microtome is used, you need a very special knife. You cannot use a blade. So the knife which is used looks like this. It's called as a microtome knife. It's extremely costly, and most important, it's very, very sharp. You need to be very careful while handling it. That's why you need to hold it only from here. And while holding it, you're not supposed to speak to anybody or just handle it carelessly. You need to place it down in a proper way once the work is done. Because the knife is so sharp that there can be a nick if you don't handle it properly. There can be a cut, and then you won't be able to take sections out of it. The cost factor is very high for this knife. It cost around that time, like 4 years back, 5 years back, maybe it was somewhere around 25,000. So you need to handle it very carefully. But it's a one-time investment. Okay, so let me show you a small video clip where you will be able to see this is the video which I have taken by a small video clip of a few seconds where I'll show you how this microtome instrument works. Before that, let me tell you that see in this instrument, this place which you're seeing, the red colored portion, that is where this knife is placed. It can be attached over there. And this portion in the center, the steel part which you're seeing, that is where your sample is placed, the wood. So the knife and the sample are placed at such an angle. That angle has to be tested properly before you begin the sectioning, so that the knife exactly slides over the sample. And then on the left side, you can see these knobs. These knobs are the ones where you can set the thickness of the section that you want. So it can be as less as 4 micrometers as well. 4 micron thick sections, thin sections in fact, ultra thin. You can get very easily if you are setting the knife and the sample properly. If you set the knob properly, you can take a section even with your eyes closed. It will come out extremely thin. That's the best part of it. And you can take multiple sections one after the other. You place the knife, you place the sample, hold the knife from here, slide it towards you. There will be a section on the knife. Take it back again towards the end. There is one more now. You have to just touch that so that the sample, if you are setting the thickness is 6 micron, the sample, because one section has already been taken, it will come up by 6 micron so that you can take the next section. Again, take it back 6 micron above. Again, take the section. You can take as many as you want. The section will remain on the knife. You need to pick it up with a brush, put it in your petri dish, and then make the permanent slides for those sections. So let us take a look at the small video clip. Okay. Observe carefully how the section is taken and it has been lifted with a brush. Automatically the knob gets moved, you can see. The section is picked up very easily. You can see from the wood a very thin section is picked up. Show once again. You need to keep the petri dish and everything ready, and if there is any debris that just falls over here, which can be cleaned later. I hope everybody enjoyed this model. Right? So that was about the sectioning part. You need to take the section, then you have to pass it across the protocol, and you have to mount it using any Canada balsam or such material, and you have to make a permanent slide so that you can view it along with the sectioning. There is one more technique that we do, and that's called as maceration. Maceration simply means that see, in a wood as an entire tissue, all the elements are present inside the vessel. You want to see an entire vessel or an entire fiber separately, complete length with the ends. You need to separate out that from the food elements. So separating out the tissues and observing them differently, separately, is for maceration. So I'll just quickly show you the flow chart for how the maceration has to be carried out. Okay, you depend across the LS, either RLS or TLS. But it has to be LS. You cannot take it transpose because you want the entire length of the fibers present. So you have to collect the wood chips accordingly, thin chips, and slightly thick wood also do that. Take the chips, put them in a test tube, proper labeling has to be done, and in the test tube we will add nitric acid. Now it is within 40% that's like the concentration that we have to work on. Sometimes people take concentrated ones, sometimes 30% or 50%. Concentrated is not chemical because the maceration takes too fast before you take it out and then fibers are destroyed. And too dilute one will take more time to macerate. So 40% was found to be a very correct one. Although the amount of nitric acid is not important quantitatively, you just need to put it in the test tube so that the wood chips are immersed in it. Then we will have to add a pinch of potassium chloride. We are separating the wood elements completely. The fibers, structures, everything. So we have to separate it out. So the cells have to be, the cell walls need to be dissolved so that they are separated. So for that, the potassium chloride is added and dissolve the middle lamella. Then the test tube has to be plugged with cotton and such shaking is done and we need to keep it as is. At the laboratory, sometimes people what they do, they hold the test tube holder and over a burner and they try to heat. That can be done, but not much advice because when you heat it, then what happens is because of the acid, the heating is too fast and that chips dissolve completely including the fibers. So that proper practical is important. That's why the better way would be to collect the material in a test tube and keep it in a test tube stand exposed to the hot sun from the outside. So there is consistent heating that takes place and nice maceration takes place. This will be better results. Across species, the amount of time required can vary. Sometimes it can be in 1 or 2 days, sometimes it can take 3 to 4 days also. But this gives very good results. So how to understand looking at the test tube that the maceration is over? The wood fibers or the chips which you added in the beginning, it might become a little whitish in color and it will settle down to the bottom. That is as a time you can take the test tube. And then you have to decant the acid and the potassium chloride fully. A little on my tin, make sure that the chips are not given out or thrown away. And then you add water, make it to the rim of the test tube, shake the test tube very nicely so that you are cleaning the extra liquid and because of the water the chips get separated very well. After you shake it well, you will see that the fibers separate out nicely and they settle down. You need to wait for some time for everything to settle down before you decant it again. This you need to repeat for 5 to 6 days. After that, after 5 to 6 times washing, carefully you will collect these wood elements with a little bit of water into a package of portions. You press it on a paper sheet and you preserve it for species identification. There are very good Herbarium present everywhere across the country and at international level also. But this is something called as a xylem. Because wood is nothing but a xylem. So xylarium is the place, as you can see in this picture, this is a place where you have preserved specimens of wood samples. So the one big xylarium that we have in our country is at FI, Forest Research Institute. Okay, so there are more than 25,000 species of wood samples, as you can see, blocks of wood, and the corresponding inventory is there, so you can access any wood sample according to your requirement and take some part of it and keep it at the right place. So that's why it's not open to general public. If you have special permission, you can access that and you can also. Okay, let me show you something more. Some more interesting facts about wood. Like we have mentioned more into the technical aspects. Like something interesting. See, wood has got very immense diversity. So wood also has got very good fragrance sometimes. We are all aware about sandalwood. It is the most sweet smelling wood in the world. Sandalwood. And this is the hardwood of the sandalwood that we use. So now we are aware whenever you are using any cosmetic product made out of sandalwood, now you should be aware that we are talking about the hardwood portion. Because that is where the deposits are present. And it is those deposits that is giving rise to the good fragrance. But nature always shows two opposite sides of the same coin. We also have a wood which is the most foul smelling, a very dirty smell. That is from Celtis cinnamomica. It's commonly known as the coating bear. Okay, if you take this and if you smell it, you won't be able to wear it. In that xylarium which I showed you, both the specimens are kept like this. So you can, if you visit sometime, you can open that, you can take the sandalwood, you will smell it, you will enjoy it. Give it back, take this Celtis cinnamomica, smell it, of course you won't enjoy it. You will also like to take the smell of this one. Okay, so that's a very interesting aspect. The most sweet smelling and the most foul smelling one. Similarly, wood has got great diversity when it comes to density. Okay, so wood is the most density and very heavy one. One of them is Hardwick cabinata, commonly called the Anjan. It's about the architecture of the internal structure. That is the differences in the density and the wall thickness of its elements. So this is the most dense one. Similarly, there is another wood which is the least density and very light one. That is called Chromalacopus, commonly called balsa. In these two specimens, in fact, these three specimens are kept over there in the xylarium. These three rocks. So this one is from Hardwick cabinata. Okay, if you go over there, you pick up that rock. It's extremely heavy. But similarly, immediately you pick up this one, the balsa, you feel as if you're lifting from a very light one. So these two interesting extreme features of one camera is a very interesting one, right? Then this fossil xylem. So now such wood species of fossils which you can get, like this one. Okay, this one is kept at the xylem over there. This is an acacia species, and it is from 1956. It was obtained in 1956 from Bihar and has been preserved. Still more ancient ones are available. Sometimes the wood powders are available, and from powder there is a lot of data can be obtained. It's a big study in itself, as I said. So these kind of wood blocks can get preserved over a long period of time and can give you a good amount of evolutionary data. That's what fossil works, right? And there are two major institutes in India where one can enroll as a researcher and you can work on wood science. One of them, as I mentioned, it's the Forest Research Institute of Dehradun. I was fortunate enough to have done a PhD from here and got good experience. And the other one, this one is actually based on entire studies on forestry, but the other one is an institute dedicated to only wood science. And first of all, there is an MSc course which is available. MSc in wood science and technology. You can go to the website of ICFI or FI and you can go to the details of the courses. Similarly, there is another one at for Institute of Wood Science and Technology, IWST. This is also kind of a sister concern of ICFI itself. As you can see, Indian Council of Forestry Research and Education, ICFI. So, IWST is an institute dedicated to wood science. They also have a xylarium, but it's not as big as FRI. FRI, there is a separate lab called Wood Anatomy Lab, separately for the study of wood science. It's another forest botany department. Okay, and FRI, because it's also a tourist place, we have a nice river museum over there. Lot of different wooden artifacts, different details about the different English species of India, the different types of materials that can be made out of. Everything is on display over there very nicely. And there's one interesting display which I would like to show all of you, because we are discussing about both things today. There is this huge specimen of a deodar tree, the trunk, which is kept on display. This is more than, we can say, 700 years old. That's the cedrus deodar, nicely polished and preserved. If you go over there and if you look at this, that is written over here. That means it was, what they can say, preserved root gas, that this plant, it must have started germinating when the Kurupna in Delhi was being built. That's how old the tree was. So a nice example of stereotypes. This is what the timber museum over there. Okay, I think the discussion for today is this much. I hope everybody enjoyed it, and if you have any questions or anything to discuss, you're most welcome. Thank you everybody for the patient listening. Yes, sir.
Thank you so much, sir. This wonderful lecture.
I think I extended my time a little more.
So, if you have any questions from the participants, you can ask. If you have any questions,
You're most welcome. My email ID and my contact number can also be provided. So, if anybody is interested to discuss, I'll be more than happy to reach out.
so we have some questions in the chat box. I'm reading them.
Sure, sir.
I think many questions were answered already in the top, and you were discussing about the growth trends and bringing the age of as well. I really find this very useful. So you talked us about the idea, like how you can identify forms of anatomical structures of wood.
Yes. This question will be very more detailed answered by tomorrow's section, because we'll be discussing about microstructure. So, I'm going to some major features in the anatomy of the wood, where I'll be showing it differently for softwood and hardwood, and then we can understand better how authentic wood identification can be carried out.
Right. So, Urvashi, 5156 asks, what kind of vessels are used to measure the growth ring width?
Okay. Growth ring width is a very apparent feature. You can use a regular normal ruler or something and you can measure it. Under the microscope also one can measure the same by in the high power or in the low power, and accordingly the calculation for multiplication can be done if you are using the optical microscope. So that's a very simple thing. There is something called stereo microscope also available. One can use of that as well. It's easy.
So 588 Kirti Adarshi has asked, if the wood may be infected, then how possible to collect such type of samples?
See, wood is generally mainly infected by fungus. So fungal hyphae can penetrate into tissues. Tomorrow I will discuss that also. That there are certain tissues in the wood which are more infected by fungus. Whenever a fungus infects the wood, it will always reach out or search for nutrients which are available. So that is mainly in the parenchyma and the vessels. So they are more infected. So apparently, if the infection is too much, then you can see that in the form of fungal growth forms. But if it is in a mild way, so sometimes what happens, you are not able to see the infection with the naked eye. So you collect it as a normal wood, and then when you study it under the microscope, you can see the fungal hyphae has grown inside. So the collection procedure means the same, even if it is an infected wood.
Thank you. The other question is from Rukmani 409. Is there any method to determine the age of the tree without
Yes. Right use of radioactivity in that case. If you don't want to cut down the tree still living and you want to find the age of the tree, radioactive methods are available where maybe some radioactive material is injected more aware of it.
Okay, so another question from Shubhani has already been answered by you. She has
Yeah,
asked
that I have already
what is
discussed. It's the separation of the wood elements which are more lignified. So in case of maceration, we don't take into account the parenchyma tissue, except that the vessels, the tracheates and the fibers.
So, okay, we have another question from Kirti.
Yeah.
What type of wood is used in paper industry?
Okay. For paper industry, as an example, I can tell eucalyptus is very commonly used, and it's like a separate department called paper and processing. So there are the bark and the wood and paper of it. But nowadays, collection of materials from the forest for making paper is quite limited because that is sort of deforestation. So recycled paper are being used. Or there are separate plantations done only for selling the other materials. Even bamboos are used sometimes. And when it comes to wood studies, even bamboos can also be a part of the study, though it is technically not a woody plant. It is a herbaceous one. Eucalyptus is the most preferred.
And this reminds me of one thing that eucalyptus is also being used to make fiber as
Yes,
cloth.
that's correct. The wood
So people from company ITC and they were discussing the cultivation of eucalyptus in India as well to produce
yes,
clothes. So there are
correct.
lots of plants in Malaysia. They are growing it there.
Exactly. Yes.
Right. So this question is
Eucalyptus is, from that point of view, eucalyptus is a very important species. But then it is very reduced in our country because it affects the biodiversity to a large
right.
extent.
Right.
It stops the groundwater and renders the other things nearby. That's very important.
So we have another question from Shubham
Yes.
470. He has asked for preserving mercury use mercury chloride. How to preserve xylithium?
The cesium chloride which is available, the fumigation of the wood samples is carried out, and then the wood pieces are preserved, which is in the form of blocks. Or we can do the polishing also. The top and then polish can be carried out on the wood and it remains as it is. Because the xylithium which I showed, it is extremely old. FRM in fact has been established by the British in our country. So that time there were a lot of British explorers who collected wood samples from other countries. In the xylithium, the photograph which I showed, we have a lot of specimens even from other countries. When I went there for the first time, there were even some names of countries which are invented now. So the specimens are based right from that time and it is still very nicely maintained. So it is done in the form of polishing and regular fumigation also.
Right. The same answer goes to you, Naveen, for the question. So Yashika has asked, what is the scope of wood science?
Okay, I think in today's lecture there are a lot of points which I mentioned where you are getting an idea what kind of scope you can create in when it comes to wood science, right? So it's about the applications in various fields. So you can do a lot of research on interdisciplinary subjects. Then, if you don't want to venture into other aspects, and there are monographic studies that one can do. So a lot of people have done PhD in that, like you select an entire family or maybe a genus. You study all the woody species of that and create a monographic data that also adds up to the study. So that research is done. And for example, in FRI, in our lab, what I had seen was there are research officers and research associates who are employed. So from everywhere in the country, depending upon the source, like sometimes there are smuggled goods which are collected. Sandalwood is one of the main example. So whenever such consignments are caught, if they are smuggled, so that wood specimen also comes to us for authentic identification. Like recently when I was there was some excavation done near Taj Mahal in Agra, and there was some wood material observed. So that was also sent to our lab for authentic identification. So the one who is employed in such labs, they have the authority to make a report and say that the sample which is provided belongs to so and so species. Like if the police is catching somebody who is smuggling sandalwood, so apparently the person might be knowing that yes, this is an expert. So that becomes a very important evidence which is accepted even in the court of law. It has got such applications, even in archaeology, sports.
And these days also people are working on one aspect which is called what do you call it? The archaeobotany is one science
Botany,
which
yes.
right.
No, it's very important. Correct.
So
So that is also very important as well. So the applications are endless.
right.
For example, I can give you in months back I was a part of a webinar. I was a participant, and this person from Pune University, he had worked on the Ajanta. That's a very important archaeological site in Maharashtra. Ajanta Ellora paintings. There are caves, rock cut caves.
If I'm not wrong.
Yes, yes, correct, correct. So there was this cave paintings, which were ancient ones. He collected materials from that which contained wood fiber. The wood science people were able to identify it based on that. So applications are so many if you just look around.
So the next question is from Saran. I think this question was self-answerable by the photographer Valerian. He's asked if the sample is very big, like you shown in the art, then how that will be preserved is alien.
See, for in Xylarium, there are big samples also which are preserved. But if it is a complete log of a wood, it is broken into small pieces. One as the Deva which I showed, then it is polished and it is better preserved in a museum. Because that cannot be available for analysis. It's a very treasurable specimen, so it just needs to be protected and preserved for display. Now, Xylarium is the one where you can collect the wood sample and you can take sections, you can take materials from, so the difference is there between the museum and the Xylarium.
So our next question is from fossil.
That's a very broad question again, because which part of the fossil is available? It depends on that. If it is the wood, then you need to understand, take sections, do maceration, study the internal structure, and then arrive at a conclusion that this is the species. But if it is wood, it can be observed.
Is it also, I think, depending on the age and type of fossil? If it is a
Exactly.
fossil,
So even radiocarbon dating is done to get an idea of the age of the fossil.
okay, so Shikha is also the same question about scope of road science and what we already discussed. Okay, of course, also can place events like gold can
So that touches upon the concept of you can say phytoremediation. There are some cases, even as a part of my PhD research, much of my reference, that my study area was the iron ore mines of Orissa. So when I collected samples from there and studied the laboratory, one aspect which I
The next
Yes. That's an ecological indicator. See, things are part of those plants where secondary growth takes place. The woody tissues are found within the growth present, but in the case of palm trees, the embryonic tissues have part of the wood is not a living tissue. But on the broader scale, if I say in the case of wood, it is only the parenchyma portion which is the living part. So you can collect wood parenchyma and do the plant tissue culture. But ultimate result is parenchyma only which you will get. You cannot get directly wood by plant tissue culture.
if you want to collect, if you want to grow these plants for wood, then obviously you have to grow these and you have to replace these saplings
Then allow it to grow.
with 20 years.
Then the
drought can change the growth rate formation.
And if it is a
Does the climate change like drought can change the growth rate
yes, yes, it will. Because when the drought conditions happen, chemical or something that is required, it just needs to be plugged in, just for physical plugging in, so that it does not remain open and no other microbial growth or any pest attack will take place from that region. That's why there is no proper mixed composition as such any chemical.
Puneet has asked me 3 or 4 years asked which are the part of human log surrounding the pitch.
Surrounding the pitch,
I don't know what
is
is referring to.
it talking about pitch?
PITCH pitch.
Pitch, I think it.
Okay, I also don't know what he's actually referring to. So he can write it again. I'm reading down.
Okay.
Another question again from Urvashi. She has asked, can radiation cause any change in the growth pattern?
Yes, can cause. And actually, there's not much studies done on this. During my PhD, my guide had suggested one thing, but it was not practically possible. That you must be aware about the Bhopal asteroid belt. So, area of the perimeter surrounding that region, in fact, even the Chernobyl disaster also you can say. In such regions, when there